Hearty greetings to you students. I am Dr.K. Sucharita, Assistant Professor, Department of Microbiology, Government College for Women, Guntur. In this video lesson, I will be discussing about isolation and pure culture techniques. As we know, microorganisms are ubiquitous in nature. That means they are present everywhere. So here, the learning objectives of this lesson is to understand the importance of pure cultures and to learn the principles of isolation methods and to understand the commonly employed methods for preparing pure cultures of microorganisms in the laboratory. So, as I said, microorganisms are ubiquitous. They exist in nature in mixed populations. Different habitats of environment like air, water and soil consists of a number of bacteria and other microorganisms in mixed populations. Even the diseased parts of plants and animals harbors a number of microorganisms. So, in order to understand their function or the role played by them in their natural situation, those organisms needs to be obtained in pure culture. So, to study every species independently, it is necessary to obtain them in pure culture. So, here an organism which is associated with a number of other organisms needs to be separated from them. This process is called isolation. So, here separation of individual organisms from the mixed community is called isolation. However, it is not easy to isolate the individual microorganisms from natural habitats and grow them under imposed laboratory conditions. For this, a great deal of laboratory manipulation is required. So, these organisms from their natural source has to be grown in laboratory medium. So, that process is called culturing. Actually, culture is a population of cells cultivated in artificial lab medium. Here, medium is food that we are providing to grow microorganisms. By definition, a pure culture is a population of cells that arises from a single cell and is free from contamination. A pure culture of a species can be prepared by isolating a particular species from mixed population and cultivating it on a suitable lab medium. If you see here in this figure, the first figure is showing a culture plate, that is Petri plate, on which you can see culture of bacteria. Here the colonies which are observed here are not of same type. You can see different kinds, and obviously there are two or more than two types of organisms, and one is lemon-yellow in color, other one is white. So, when such bacterial culture is observed under microscope, you will see different kinds of cells, and that can be seen in this microscopic field. Here you can see rods, cocci and different kinds of organisms, and this microscopic photograph also showing two types of organisms. It is a gram-stained picture. Both these are gram-stained pictures. Here you can see two different organisms. One is gram-positive organism, second one is gram-negative organism. So, this kind of presence of more than one kind of organism in a culture is called mixed culture. So, in that way, microorganisms are present in natural habitats. Whereas, here you can clearly see that this plate is showing only one kind of colonies. When it is observed under microscope, again you can see all the cells are of similar kind. So, this is pure culture. Pure culture is also called axenic culture. Why it is important to purify microorganisms? Initially, the pure culture techniques were developed by Robert Koch. These are so important that the development of pure culture techniques by the German bacteriologist Robert Koch transformed microbiology. Within about 20 years after the development of pure culture techniques, most pathogens responsible for major human diseases had been isolated. So, it is important to have a pure culture of microorganism to understand the role played by the microorganism in nature and to study the characteristics of one species. Here one kind of organism is called one species. when a pure culture is also referred to as one species, because pure culture is arised from a single kind of cells and all the cells are identical. For definitive identification and characterization of organisms, for this purpose we need to understand the growth conditions and nutrient requirements of microorganisms. And once organisms are obtained in pure culture, we can also analyze the metabolic products it produces. And it is also important to determine the causative agent of disease. Particularly in field of medical microbiology, sometimes it is important to identify the organism causing the disease. For that purpose also, the sample isolated from sample taken from patient is analyzed, it is used for pure culture techniques. So again, large population of pure cultures are required for several microbiological processes like antigen preparation, antibiotic sensitivity assay, typing bacterial isolates, etc. Then we will go to the methods of isolation. As microorganisms show great physiological variation, these properties can be exploited to isolate specific groups of microorganisms. Before the advent of different culture media, natural sources like solid fruits and cut potatoes were used to isolate microorganisms from different sources. But with the introduction of successful culture media, a variety of techniques have been developed for the isolation of microorganisms, particularly bacteria. However, each technique has certain advantages and disadvantages of its own. Hence, no single isolation method can be commonly used for all types of microorganisms. The effectiveness of any isolation method actually greatly increased by using special media. So, what is this media? Actually, a culture medium is a substance or preparation used to support the growth of microorganisms. So, it should provide nutrients required for growing microorganisms. So, usually this culture media is of three types: solid media, semi-solid medium and liquid medium. So, liquid medium is also referred to as broth, and a liquid preparation having nutrients is called liquid medium. This is generally used for inoculum preparation, blood cultures, etc. An example for liquid medium is nutrient broth, that is routinely used in microbiology laboratories. Then solid medium, this is also called solid agar, is prepared by adding agar, a complex polysaccharide derived from marine algae, to liquid media. This is used for obtaining pure cultures and colony morphology, pigmentation. So, here we are preparing solid media by converting liquid media into solid. The agar is here, once agar is added to liquid media and it is melted at high temperature of the autoclave, then it remains liquid. When it is cooled down to a temperature of about 45 degrees Celsius or below to that temperature, it gets solidified. Then once a medium is prepared, that medium is inoculated with microorganisms. Actually, inoculation is a process where microbial culture is transferred to a fresh medium. For this purpose, we need inoculum. Usually, inoculum is a suspension of microorganisms from natural sample or something, anything which is having microorganisms. So, here inoculum is the sample from which microorganisms will be isolated or transferred. For inoculating fresh medium, we use some devices. One of that is inoculation loop. Here you can see inoculation loop. It is a tool usually made of platinum or nichrome wire. This wire is made up of platinum or nichrome, in which the tip forms a small loop with a diameter of about 5 mm and is used to smear, streak or take an inoculum from a culture of organisms. Once inoculation is done on fresh medium, then the medium should be incubated. Here incubation is, we are providing proper conditions for growth and development of organisms. So, all the physical parameters at which you are incubating comes under incubation conditions. Then while working with microorganisms, you need to maintain aseptic conditions. It is a must. Why these are required is to prevent contamination of culture with environmental microbes and preventing contamination of yourself or the environment with organism in the culture, and to remember everything is contaminated with a variety of environmental microbes. Once agar is prepared, it has to be melted, and that agar has to be poured into petri dishes, and these are the culture dishes routinely used in laboratory for preparing solid media. So here molten agar is poured into plates. Once it is poured, they are allowed to solidify. After agar solidifies in the plate, you can see a solidified agar plate. This is generally called nutrient agar plate, where medium is present. So petri plate is a shallow circular glass or plastic dish with a loose fitting cover over the top and sides. You can see two halves, bottom half and upper half. So upper half overlaps the bottom half, and this is used for culturing bacteria and other microorganisms. So solid agar medium is more suitable for isolation of microorganisms because they can trap and fix cells and lead to development of a colony on a petri plate. So here if you see, if a mixed culture is placed on agar plate, all the cells are spread on the plate and each cell will multiply, and after incubation, when sufficient time is given to that, they will develop into a macroscopically visible growth. So initially cell is not visible, but only after it multiplies several times, it will produce millions of cells, and all those cells are clustered together, and those can be seen as a mass of cells. Those are called colonies. So if a mixture of cells is spread on agar so that every cell grows into a completely separate colony, a macroscopically visible growth of microorganisms. Each colony that is formed on the agar plate represents a viable cell present in the original sample. So by this way you can separate organisms mechanically on the plate. Then there are several ways to prepare pure cultures. Few of the more common approaches are reviewed here. First one is serial dilution, enrichment culture, plating techniques which include streak plate, pour plate and spread plate, single cell isolation methods, capillary pipette and micromanipulator, then use of special selective medium. First we will see serial dilution. Joseph Lister in 1857 obtained pure culture of bacteria using a serial dilution in liquid media. He took a fluid containing mixture of bacteria and diluted it with sterile medium. Then he transferred the diluted sample into a nutrient solution. Upon incubation, he found that a population of single kind of organisms identical to parent cell have been grown into that culture. All of these organisms were of similar kind, so hence he was the first person who successfully cultured organisms in pure culture by using liquid medium. In practice, serial dilution method proved to be tedious, difficult and uncertain for routine use. The microbes that are predominated in the original mixture can be isolated. So all the organisms present in the mixture cannot be isolated because only the predominated microorganisms can be found in highest dilutions and they can be isolated. Then coming to serial dilution, actual serial dilution involves diluting a sample serially in a series of tubes. So if you want to isolate a sample for 100 times, take 1 ml of that sample and add 99 ml of sterile medium or saline to that. So you get 100-fold dilution. But in natural sample usually cells are very large in number and about 10 to the power of 7 cells are present in natural sample. So all of them cannot be diluted, all of them cannot be plated on an agar plate. If they are plated on agar plate, you will see a crowded appearance. So in order to prevent that, organisms are serially diluted. For serial dilution, first sample is placed in, so we have to take series of tubes, each tube containing 9 ml of saline medium. To the first tube add 1 ml of sample and mix it thoroughly, so that this 1 ml will be distributed into the 9 ml, total will be 10 ml. So, this 10 ml sample is now holding the original number of samples that are present in the sample. Suppose it is having 40,000 number, 40,000 number of organisms are diluted into 10 ml. So, we have added 1 ml containing 40,000 ml of cells to this 9 ml, and now it becomes 40,000 cells in 10 ml, that means 4,000 cells in 1 ml. From this, take 1 ml and add it to another tube having 9 ml of saline medium. Then what happens? This 1 ml will get mixed with 9 ml. It is diluted 10 times. Similarly, you take 1 ml further and dilute it into another tube. Again from this, mix the contents thoroughly, transfer it to another medium and mix it. Finally, in the final dilutions, you can see very few numbers. So, we have started with 40,000 number, but here they are 4,440, and finally, theoretically it should be 4 in this tube. First dilution is referred to as 10 to the power of minus 1 or 1 by 10 dilution, and the second one is 1 by 100, third one is 1 by 1000, and fourth one is 1 by 10,000. Then the last one is 1 divided by 100,000. So, it can also be written as 10 to the power of minus 1, 10 to the power of minus 2, 10 to the power of minus 3, 10 to the power of minus 4 and 10 to the power of minus 5. So, here when these samples are plated onto agar medium, you can see number of colonies. Number of colonies represents number of viable cells present in the dilution. So, by doing this technique, we can estimate the number of organisms present in the original sample, and for that we have a formula. That formula is number of cells per ml of sample is equal to number of CFU divided by volume plated into dilution plated. Here number of CFU, if you take this as example, this is 10 to the power of minus 4 sample, 40, we will take this, 40 divided by 0.1 ml into 10 to the power of minus 4 is equal to 40 divided by 10 to the power of minus 1 into 10 to the power of minus 4, that becomes 4 into 10 to the power of 5. So, those are the number of cells present in original sample.